Hi, everyone. I would like to start with thanking Dr. Paul Boutros and for joining us today and welcome him to our session. Um, Dr. Boutros earned his BS degree from University of Waterloo in chemistry in 2004 and his PhD from University of Toronto in medical physics in 2008. He also did an MBA at the Rotman School of Management, also at Toronto, and he's been actually performing his independent career, starting with Ontario Institute of Cancer Research, starting as a fellow in 2008 till 2010, and then he continued there as a principal investigator 2018. Since 2018, Dr. Boutros has been at UCLA, and it's my pleasure today to welcome him to our Canary series. The stage is yours, Paul. Thank you very much.
Thanks so much for having me. Just sharing my screen. Excellent. Can you guys see and hear it?
Yes, I can see it and hear it.
Excellent. Well, I guess you hear me, not the screen, but okay. So it's excellent to get a chance. I'm so sad I don't get to come down and see you guys. As Utkan mentioned, I moved to UCLA just about 18 months ago, a little bit more than that now. And so I'm going to talk about a mix of stuff that I did since I moved here and stuff that I did before. And so the core idea of, quick disclosures, I'm on 3 scientific advisory boards, none of which have anything to do with what we're going to talk about. So, the core of what we're going to talk about is this idea of localized disease and detecting which localized diseases our cancers are going to be aggressive early as possible at initial diagnosis or even before. And so we'll spend a fair chunk of time talking about DNA, then getting into the central dogma, thinking about imaging, both cellular and histologic, and cellular and radiologic, sorry, tumor evolution, and then ending up on the host and thinking about how we can leverage all of these things together. So yeah, I'll go back almost exactly 16 years. This is the first time I ever saw a Kaplan-Meier curve. I saw this on the second day of my PhD. I'd never seen one before. And this is for early stage lung cancer. Doesn't really matter what tumor type it is. Obviously, you see on the y-axis the fraction of patients alive, and on the x-axis how long they've been alive. And we were talking about this in the context of chemotherapy for early lung cancer. And I stared at this plot for a few minutes and looked at it and then said, wait, I have to go backwards. Why is there so much variability? Like, why are there some patients who are having a relapse of their disease, or in this case, mortality of their disease, really early after diagnosis, and other people who have long-term good outcomes? And the clinician who we were working with at the time looked at me, waited a beat, and said, we have no idea. This is like basically an unanswered question. And so that's the question I've been interested for the last 16 years. At diagnosis, how can we understand what are going to be the differential outcomes so that we can do a better job of managing early cancer when it's still curable, where we have this group of patients where in almost every cancer type, we have options for intensification of therapy, sometimes more intense radiotherapy or adjuvant courses of chemotherapy, or even adopting targeted immunotherapies earlier in the disease course. And of course, there are this group of patients who have long-term good outcomes, and in many cancers, breast, prostate, thyroid, are potentially being overtreated and suffering the morbidities and financial costs of treatment without really deriving benefit from it. And so, that's a decade and a half that I've been really thinking about this problem and how to do it well. And pretty early in that process, about 12 years ago, I chose to put a substantial amount of focus into prostate cancer. And about a decade of my life has been thinking about it in this context. And the reasons to study prostate cancer for really triaging of aggressive early disease are 3. First, it's a high incidence tumor. Depending on where you are, between 1 in 7 and 1 in 9 men will be affected with it over their lifetime. So lots and lots of diagnoses, about 400,000 a year in North America. It only kills about 1 in 4 or 1 in 5 of those individuals. So you've definitely got a subset of aggressive tumors, and a very large one. You know, it's 100,000 people a year, but it's still not the majority of cases. And that leads to a real balance between incidence and mortality that's important to understand. And then third, if you take a look at this plot of incidence, you can see in the early 1990s, there was almost a doubling in the rate of incidence in North American populations. And this corresponds to the introduction of PSA testing. It's a simple blood test for PSA, a serum antigen. And when it's present, it's very sensitive, but not very specific for detecting prostate cancer. You can see that this almost doubling in incidence and diagnosis was not really accompanied by any change in mortality, and the shallow decline over here is largely a function of improved surgery, radiotherapy, and extensions in the lifespan of patients with metastatic disease rather than improved cure rates up front. And so given that, you might think that we struggle to figure out how to prognose prostate cancer patients, and you'd be exactly right. So standard of care today is to use that blood test, PSA, linked to a biopsy. And of course, a biopsy in prostate cancer is taken transrectally. And that means that it's got a very high 2 to 3% chance of infection. And in fact, 2 to 3% is the chance that a man will be sent to the emergency room with sepsis. So these are really, really morbid procedures for something that's being done quite frequently. And this radiology is not yet standard of care for diagnosis of localized prostate cancer. Instead, we use a digital rectal exam where a physician inserts a lubricated finger into a man's anus and palpates the prostate, trying to look for a bump that tells you about the presence and extent of the disease. And that's kind of it. Multiparametric MRI is on the cusp of standard of care. Isn't quite there yet, but probably will be in the next 5 years. And that leaves a lot of problems in how we diagnose aggressive from non-aggressive prostate cancer. And maybe the most inspiring thing I'll show you is that those 3 pretty simple clinical tests, a digital rectal exam, serum PSA, and Gleason grade, so grade of tumor on a biopsy, do a remarkably good job of stratifying people into low, intermediate, and high risk of relapse at time 0 of diagnosis. And when I decided to start focusing on prostate cancer, I decided to start focusing on intermediate risk disease. It's a perfect classification problem. About a third of these men are eventually going to have a metastatic relapse of their disease, so they're undertreated. About a third, we know from cohort studies and autopsy studies, probably have a tumor that will never kill them, and they should never have been treated. And a third are being cured by existing therapy. And that existing therapy is dependent on the risk groups. For the low risk individuals, it's just monitoring to see if the tumor progresses. For the intermediate risk, it's definitive local surgery or radiotherapy at equipoise with curative intent. And for the high risk, it's definitive local plus adjuvant in some cases, not all. And the adjuvant is typically, but not always, hormone therapy targeting the androgen receptor. So we decided to focus on intermediate risk localized therapy. We put together a large national cohort. This went across the nation of Canada, so 3 provinces. And we had pre-radiotherapy biopsies, pre-surgery biopsies, and surgical specimens, all linked to long-term outcome, centralized pathology. And then we did all the genomics you could like to try to predict which men were going to have a disease that would relapse after local therapy and which ones would not. That's kind of the focus of the next few minutes of the talk. Okay, so based on that, then we said, let's start off looking at the DNA. Cancer is an evolutionary disease, so this should be straightforward. And the initial idea we had was, if we're going to look at the DNA, then we should think about the tumor heterogeneity and the tumor evolution, because you have a prostate which is the size of a walnut, and if you're only looking at a small sample of tissue taken from a needle biopsy, you have to make sure that that represents the part of the cancer that you care about. And so we used what's called whole mount prostatectomy specimens. So what we've done is take the prostate the size of a walnut and then serially slice through it. You can think about putting it through an egg slicer. And what you're seeing here are individual layers of that egg slicer, one at a time. And for each layer, our pathologist would go through and mark it and use laser capture microdissection, or in a few cases, manual macrodissection to say, okay, here's a region of Gleason pattern 4 cancer. We will extract that and do DNA whole genome sequencing. Here is a region of mixed Gleason pattern, just Gleason pattern 3 cancer. We will laser capture microdissect this and do DNA sequencing of it. And so we did that for a series of different prostate cancers, about 15 of them. And this is a complicated plot. I'm just going to get you to focus on this green rectangle in the middle. Each of these columns is a different spatially separated region from a single man and a different region of his prostate cancer. And this bar plot on the top is telling you the number of mutations in protein coding genes. And you can see it ranges from about 20 to about 150. So roughly an order of magnitude variability in the number of genes mutated from region to region within a single man's prostate cancer. Okay, here you see 5 regions from a different man's prostate cancer. And again, you see this order of magnitude variability. So it's a profound difference. And of course, these are protein coding point mutations, but the same thing is true if we look at copy number aberrations, or if we look at genomic rearrangements. Actually, it's true if we look at the epigenome or the transcriptome or the proteome as well. Effectively, there's this large variability in the number of mutations of any kind from region to region in a tumor. And so you could fairly say, sure, Paul, tumors have variability spatially, but who cares? It's not necessarily going to affect what I care about understanding aggressivity. And so let's take a look at that first man we talked about, where we had 9 regions out of his tumor. You can see here those 9 regions. Each of the rows is a different region. They're linked to the anatomical, histological, and immunohistochemical features of those regions of the tumor. And I'm just going to blow out a tiny part of this story, this cancer-related genes panel. So you can now see those 9 regions laid out as columns. The first one is the diagnostic biopsy. So this is the one that was used to the index lesion to make clinical decisions. And you can see here that there's a green rectangle for PIK3CA. So that's a protein coding point mutation in PIK3CA. It's H1054. So it's druggable, and this would be one of the very few prostate cancer patients with localized disease who might be a candidate for a basket trial. You can see down here for P53, there's a blue dot. That's a heterozygous deletion of P53, deletion for the dot, and you know, that's an adverse prognostic factor for radiotherapy, although not a very strong one. So you have a bit of a story here. This man might be a candidate for a basket trial, and you might weakly prefer radiotherapy to surgery. The problem comes when we take a look at the rest of the tumor. So you can immediately see that the PIK3CA mutation only occurs in 1 of the 8 surgically resected regions of the tumor. The P53 deletion is on only 3 of 8. There's a BRCA1 homozygous deletion, which is just an adverse prognostic factor period, which is in 4 of 8 regions of the tumor. If we now superimpose upon this the pathologic aggressivity, so the fraction of high-grade disease, it turns out that the BRCA1 deleted regions are the most aggressive. And now if you ask, okay, but which of these actually gives rise to the relapse? Unfortunately, this man did have a relapse of his tumor, and it was the clone right over here, which had none of the mutations that I just spent 90 seconds telling you all about that gave rise to the relapse. And so, you know, that's a problem. If we can't figure out from the biopsy or from multiple biopsies which is the most aggressive region, especially looking at individual genes, how are we supposed to come up with a good biomarker? And so that led us, and us here is Rob Bristow, a radiation oncologist who I worked with for a lot of these studies, and Emily Lalonde, a brilliant PhD student with us back then, who's now faculty at Wayne State. We started saying, okay, if individual genes are not going to do it, then maybe what we need to start thinking about is processes. And so we asked, what is the type of mutation that occurs earliest in prostate cancer evolution? And the answer was copy number changes, large-scale gains or losses of part of the genome. And we clustered that, just did simple subtyping. And the subtypes look clearer here, where you can see a subtype characterized by gain of chromosome 7, loss of AP and gain of AQ, loss of AP and loss of 16q, and a kind of quiet subtype. Sure, they're convincing, and there are some driver genes in each of these, but what's really interesting is if we superimpose this on terms of clinical outcome. And you immediately see the red subtype has good outcome, the yellow subtype is late relapsing, and the green and blue have a poor outcome. And what's interesting is this red subtype, it's cluster 4. Tumors that don't have mutations have better outcome. That sounds obvious to you. It sounds obvious to you, and I know it because as soon as I saw it, I said, this is obvious, this must be in the literature. It is kind of. Genomic instability is bad, but there's actually only one other tumor type which shows as strong a trend of genomic instability with outcome, and that's breast cancer, which is fascinating in a whole series of ways that breast and prostate would be similar in this way. Lots of other cancer types show associations, but weak ones. And so that means, you can come up with a biomarker without thinking too hard. You take the percentage of the genome altered, you just add up the number of bases in the genome that have a copy number aberration, and look at that. It's not very sensitive, but it's really specific at telling you which prostate tumors are going to have a relapse. And the threshold here, 7.5%, corresponds to really just a couple of chromosomes in the genome. So all of these prostate cancers that are quiet and have few mutations really are indolent. Not all of them, but most of them are indolent. Okay, so yeah, why is there that much variability in the amount of mutations present in a prostate cancer? We weren't sure, and so we spent a fair bit of time thinking about that. And we came to the conclusion that it might have to do with hypoxia. Hypoxia, molecular oxygen, is highly variable in prostate cancer in a series of reports, is associated weakly with patient outcome, and so we thought it might be associated with genomic instability by abrogating DNA damage repair pathways. And so we looked at a series of 160 patients where we had copy number measurements in the biopsy and used a needle electrode at the time of biopsy to get physical measurements of oxygen content. And the correlation here between the oxygen content on the x-axis and genomic instability on the y-axis is, well, non-existent, right? There's really no association. Once we've scaled this up to larger cohorts of 600 or 700 patients, we see a tiny association, but really not so relevant. So hypoxia and genomic instability are not correlated, but what's really remarkable, almost magical, is that when you put these two things together, they are synergistically associated with patient outcome. Here at the bottom are tumors where they have both high levels of genomic instability and low levels of molecular oxygen, highly hypoxic. And at the top, it's the inverse, patients who have normoxic tumors and have relatively few genomic aberrations at the copy number level. And so this tells you that it's sometimes not about the single process, but multiple co-occurring processes. I'll come back to this again in about 15 or 20 minutes after I show you a whole bunch of things that don't work. So, the first thing that made me wonder is, okay, if we can take a look at hypoxia and it is associated with genomic instability very, very weakly, does it tell us something about the types of mutations that accrue in a tumor? Because, sticking a needle electrode into the prostate at every biopsy is not a clinically translatable assay. So a graduate student on my team, Vinny Bandari, went ahead and found a way to be able to map hypoxia directly into the TCGA cohort using a series of different validated RNA signatures and things that we've used both FASA imaging and PIMO as a molecular assay in immunohistochemistry. And that allowed us to measure individual cancer types from the least hypoxic, thyroid and prostate, to the most, lung, squamous, and head and neck cancer. And maybe the most important thing to say is, sure, cancer types vary in how hypoxic they are, but look at the spread from individual to individual within a cancer type. Within any tumor type, some tumors are partially, non, or completely hypoxic. And hypoxia leaves a very clear scar on the tumor genome. So here we're looking at kidney clear cell, tumors laid out from the most to the least hypoxic. And you can see on the left, highly hypoxic tumors are massively enriched in deletion of CDKN2A and gain of MYC. And you can definitely say VHL signaling means that kidney clear cell might be a weird tumor type. So let's take a look at breast cancer. Here we're looking at luminal breast cancers. You can see that P53 point mutations are enriched in hypoxic breast cancers. PIK3CA point mutations are enriched in normoxic breast cancers. MYC amplifications in hypoxic breast cancers. This occurs in about 2/3 of cancer types. So that actually means you can infer the characteristics of hypoxia just by looking at the mutations present. There are signatures and very, very strong features that tell you what's there. And so we incorporated that into a biomarker that can use 100 genomic regions to infer simultaneously both hypoxia and genomic instability. It's about 80% accurate, pretty sensitive, not really specific in our initial validation cohort. And of course, kindly reviewers asked us for a large validation cohort. So we worked with GenomeDX, a company in Vancouver, to look at 1,000 patients. And you can see here, the performance of our signature is an AUC here of about 0.8. Current clinical variables are an AUC of about 0.6. And the rest of these black lines are currently used clinical or academic biomarkers. And so, you know, an AUC is pretty good. Actually, an AUC, and this is like the only time in my life the validation cohort will do better than the initial validation cohort. You can infer it as we're making 20% of errors. And so, you know, the next 15 minutes of this talk, before we kind of return back to the idea of multiple processes, is going to be really themed around why are we missing 20%? What's going on? Well, the first thing you might think is, okay, maybe this is related to the risk group that you look at. And so we went ahead and did validations in low, intermediate, and high-risk patients, each about a couple of hundred patients, and we see that same AUC, so it's not obviously a feature of Gleason grade or something like that, but instead, we're still making 20% of mistakes, so we need to kind of track them down. Okay, good. So what might we be missing? Well, the first obvious thing is we looked at copy number. If we looked at copy number alone, the rest of the information is probably captured in translocations, rearrangements, point mutations, stuff like that. So we took a set of 200 of those tumors, we did whole genome sequencing, array-based methylome and RNA profiling, and we looked for driver mutations. And we found a whole series of interesting things. I'll just point out one to you. This top panel is taking a look at the number of point mutations in the genome. And notice this subset of tumors with almost 0 point mutations. These are cancers that have effectively no mutations in their genome. In many cases, no mutations at the point mutation level, no mutations for genomic rearrangements, and no copy number changes. It's hard to understand how these things are even cancer given how few mutations they have, but some of these even relapse and kill the patient. We put those information together and we identified a set of 40 prostate cancer driver events. They're laid out here as the rows. And we used those together in a machine learning model to try to predict patient outcome. And we came up with a biomarker that has an AUC of about 0.8. So if you think about it, an AUC of 0.8 is exactly the same as the AUC that I had from my 100-region nanostring-based assay to look at hypoxia and genomic instability. It's not additive with hypoxia, and this one would cost 10,000, and the nanostring assay would cost 200, so that didn't get us anywhere. So fine. If it's not DNA-based mutations in the nucleus, it must be DNA-based mutations in the mitochondria. So we went ahead and looked at the mitochondrial genomes, that's the circular genome here, and each of these peaks is a recurrent somatic mitochondrial mutation present in prostate cancer. So surprising at how incident those are, several as high as 10% of cases. We looked at how those mitochondrial mutations, laid out here as columns, associated with nuclear driver mutations, those 40 that I mentioned before, laid out as rows. And every time you see a black background with a red or blue dot, that's a case of a nuclear mitochondrial interaction. So where a specific nuclear mutation is significantly more in red or less in blue, likely to occur in the presence of a specific mitochondrial. So actually, you can't fully understand what's going on in the nuclear genome mutational space without knowing about the mitochondrial. And in a few cases, this actually shows synergistic effects on outcome. So the red curve here with really poor outcome are patients that have both mutations in MYC, amplification of MYC, and in the mitochondrial origin of replication. And in blue, one or the other, and in black, just one. And so you can immediately see an interaction where this is much worse, only affecting a few patients. This explains none of the missing 20%. So unfortunately, that was not helpful. Okay, fine. So if it's not DNA, then we need to just start walking through the central dogma because cancer regulation is complicated. And so obviously, if it's not genetic, it's epigenetic. So we went ahead and looked at 620 methylomes from tumors, many overlapping with our whole genome sequencing. You can immediately see at the top, these are the mutational characteristics of them, and we see all sorts of interactions where specific mutations are highly enriched in specific epigenetic subtypes. We link that to enhancer profiling using H3K27 acetyl-ChIP-seq in a couple of papers, put that together into a biomarker, which you can see the Kaplan-Meier for here. I'm showing you a Kaplan-Meier. Actually, the AUC for this is about 0.7, so going in the wrong direction. Okay, not epigenetic, so it must be transcriptomic. We went ahead and did what we call ultra-deep RNA sequencing. So this is 1 billion RNA-seq reads per sample. Almost as soon as we did so, we identified a large series of reads which weren't properly mapping. And those reads that weren't mapping turned out to be from circular isoforms. We were pretty surprised at how many circular isoforms we had in individual tumors. So working with Hansen He, who's a molecular biologist back in Toronto, we developed a methodology to be able to detect these from our RNA-seq data with high fidelity, and then B, a dropout screen where we would come up with shRNAs targeted specifically at the circular molecules and not at the linear forms of those genes. And these are just like circularizing through a backsplicing event. And when we do so, we identified a set of circular RNAs that are essential to prostate cancer cell line growth, which is kind of crazy. The linear isoform is not essential in purple, but with multiple hairpins in the orange and the orange and the green, these were essential or at least significantly associated up here. Several of these actually produce peptide products, which is crazy, and a few others we were able to functionally validate serve as microRNA sinks or sponges or shuttles. Has nothing to do with outcome. A biomarker based on DNA, RNA, and epigenomics has an AUC of 0.8. Okay, so it's not the epigenome, it's not the genome, it's not the transcriptome, it must be the proteome. So working with my good friend Thomas Kisslinger, who you will have coming to speak with you guys in January, I think, well, not coming, but you'll get to hear him speak in January. We looked at the tissue proteome of 75 tumors. You'll notice our sample size keeps dropping. We both started to run out of money and realized to find out there's no signal, you don't have to do 600 patients. You can easily see that there's no signal off of about 100 patients or so. So for these tumors, we had the full stack. You know, we had DNA, RNA, epigenomics, proteome. And we discovered a series of pretty interesting things from the prostate cancer proteome. First, we wanted to see if there was a difference in the genes that were associated with phenotypes at the protein and the RNA level. And so the first thing that we looked at was the most common driver aberration in prostate cancer. It's a fusion gene, ETS fusion. TMPRSS2-ERG is the most common, accounting for about 90% of these fusions. And we looked at the association at the protein level of genes that were different between ETS positive and ETS negative, and at the RNA level. And you can immediately see a good correlation here. Spearman's rho is about 0.7. But it's actually not that good, because if you take a look at the axes, the effect sizes are really, really different. So just to take an example here, relaxin-1 is changed about 8-fold at the RNA level, but 2,000-fold or so at the protein level. Even here, if we take a gene like ALOX, it's changed about 20-fold at the RNA level, but about 40-fold at the protein level. And of course, there are outliers like ERG, which is basically unexpressed at the protein level, but present at the RNA level basally. And so, that dynamic difference in protein belies real differences in the pathways that are changed. This is just your typical pathway analysis where each node is a different pathway, and we've kind of clustered them using an unsupervised algorithm. And if the node is colored on the left, it's associated with ERG status at the RNA level. And if it's colored on the right, it's associated with ERG status at the protein level. And you should immediately note that there is not a single node that is colored on both sides, right? The pathway correlates of RNA and protein are entirely different. And this really, really, really made me start to be skeptical about my own RNA sequencing data. If it so poorly reflects what's going on at the protein level, how much do I really care about the pathway analysis that I do on RNA-seq data in a big lab like 5 times a week? Not that much. And, it makes you wonder just how tightly associated abundance of protein is with abundance of RNA. And so we grouped proteins into deciles. This is the 10% most abundant protein. This is the 10% least abundant protein. Each point is an individual gene, and this is the Spearman row between RNA and protein. And so you can see for the most abundant 10% of proteins, the median correlation is 0.3. So if you think about that, that's correlation. So that means that RNA explains 0.3 squared, about 10% of the intertumor variability in protein level. So RNA is actually not all that informative about what's going on the protein level, and this made me even more skeptical of RNA-seq datasets that we were working with and continue to work with, because it's just not that tightly associated with protein data. We were pretty skeptical of this result, but fortunately CPTAC was able to validate large fractions of this in several other tumor types around the same time. So there have been papers in breast, lung, colon, ovarian, head and neck, and kidney now at least, and I might be missing a few. So this is just a general phenomenon that RNA is not a good explanatory feature for protein. And so, this gives you the first hint of something that actually works. If you take a look at the genes that are associated with patient survival at the RNA level on the x-axis and the protein level on the y-axis, you basically see a scatter plot with a cloud, nothing in there. There are a few genes that are correlated at both levels, and we validated a few of these, but more genes are these outliers like MDK, which is associated with outcome at the RNA level but not protein, or GLUD1, which is associated with prognosis at the protein level but not the RNA level. And if we add protein to our existing biomarkers or construct multivariate ones, we managed to improve the AUC from 0.8 to just under 0.85, which is better, but not all that much better. And so, at this point, I was actually a little depressed because we tried everything I could think of, and clearly there was no obvious molecular feature that was working. And so I went for a series of chats with colleagues that were really deeply engaged in prostate cancer diagnosis. And that meant radiologists and pathologists. And we'd go for beers, and I would basically just kind of pick their brain for what they were seeing and what they thought maybe we should be thinking about differently. And, the first thing that happened when I talked to our study pathologists was, I think you should look at these sub-histologies called intraductal carcinoma and Cribriform architecture. So IDC, as it's called, is a histopathological sub-histology that's part of prostate cancer diagnosis. These two regions of adenocarcinoma, or invasive carcinoma here, and IDC here, are both Gleason pattern 4. They're the same grade, but clearly they look really different. And so the question was, do these look different molecularly? There's been several hints that these have differential outcome. And so he said, excellent, let's take a look at this in our cohort and a series of other large cohorts and see if we can trigger some understanding of whether or not this has some relationship to our struggles to come up with good biomarkers. And so pretty obviously, IDCCA, intraductal carcinoma, Cribriform architecture, is associated with worse prognosis. That's not a great biomarker by itself. It's an AUC of about 0.7, but it's a big effect, and it has nothing to do with anything other than a pathologist read of the H&E, and this is actually reported as standard as part of the updated ISUP pathology grading now. Okay, great. Well, what does that mean if we take a look at individual tumors in either paired analysis, where we laser capture microdissect the regions, or look at tumors that do and don't have IDC components? You immediately see that the genomic instability of these IDC tumors are bigger. And so, this made me wonder a whole lot about whether this has something to do with whether they show up in radiologic assays. And so having a couple of drinks with some of my radiologist friends, they immediately said, well, there's a surprising fraction, about 30% of localized prostate cancers that can't be seen under multiparametric MRI. I wonder if these have anything to do with MRI visibility and intraductal carcinoma. And almost immediately you see a big, big enrichment for intraductal carcinoma tumors in MRI visible tumors. And MRI visible tumors are more aggressive, so that's really concordant. And in fact, MRI visible tumors are also more genomically unstable, MRI visible versus invisible tumors. And I'm not showing you, but they're also more hypoxic. And so you start seeing maybe this idea of multiple conflating features are coming together. And maybe part of my problem in looking at molecular features is just that like tumors are changing a lot, and I need to start thinking about this a little more holistically. And so that's what we started doing. We started saying, let's start linking these different data sets together. We did proteomics of MRI visible and invisible and identified a large set of proteins that are associated with that. And that led us to say, okay, we need to go backwards and stop treating prostate cancer like it's an entity. We're treating this a little bit like it's a snapshot, but actually it's a video. And we need to figure out how to at least infer previous frames of that video. And so that led us to start thinking about tumor evolution. And I had no idea how to be able to do that, inferring going backwards. So we put together a crowdsourced benchmarking challenge. We put together, working with friends at OHSU, the Crick, the Sanger, University of Toronto, Memorial Sloan Kettering, a bunch of ideas of this is what an idealized tumor evolution should look like. Now, how do we solve this from a whole genome sequence? We were able to come up with a way to simulate those tumors, and as part of it, we found a number of algorithms that worked really well in doing that. So we put those algorithms to the use on our own data set, and we applied it to a set of about 300 localized prostate cancers. And so these are the evolutionary trees. This first one here is 119 tumors that appear to be monoclonal in the index biopsy. Obviously, if we looked at larger fractions of the tumor, we'd find spatial variability, but in the index biopsy, they are monoclonal at the resolution that our sequencing assays give us. And you can see the branch lengths based on the number of mutations and a single population. Here we see a subset of tumors, about 170, which are polyclonal with multiple subpopulations, sometimes linear and sometimes branched. I'll actually come back to these branched tumors in a couple of slides. Well, one thing that happens when you do this is then you're able to time individual mutations, and you can ask which mutations occur first and which ones occur later. So you can start looking at mutational processes. So for example, this gold mutational process, which is decreasing in importance over evolutionary time in multiple cancers, well, that's the age-associated deamination profile of point mutations. Equivalently, this red process, which is rising in importance over evolutionary time, that's HR deficiency. So tumors are becoming more and more driven by mutations associated with HR deficiency and less with stochastic ones associated with just plain old DNA replication, at least in prostate cancer prior to diagnosis. And so now we can start putting these things together, and we can come up with a biomarker that incorporates all those features that I told you about. And so this top group, in 8 years with less than a 5% relapse rate, these are tumors that are monoclonal, normoxic, genomically stable, MRI invisible, and sorry, typo, IDCCA negative. They don't have that sub-histology. And at the bottom, with a 70% relapse rate, those are tumors that are polyclonal, hypoxic, genomically unstable, MRI visible, and IDCCA positive. Sorry, I inverted those. And then in the middle, breaking out according to basically a counting exercise, are tumors that have a subset of those features. And you can immediately see there's not that many tumors in the middle. Tumors tend to have multiple of these or none of these, or sorry, multiple of these on the bottom and none of these. Actually, what does this kind of look like? It looks like that very first survival curve I showed you of low, intermediate, and high-risk prostate cancer. But at time 0, clinically, all of these were intermediate-risk prostate cancer. This biomarker has an AUC of 0.95, and we're in the middle of a large-scale validation of it, about 1,000 patients to really confirm that in a retrospective cohort before we start looking prospectively. Okay, so we call this Nimbosus, a gathering of stormy clouds, with the idea that multiple co-occurring aggressive features make a tumor really bad, and that we need to think about it in terms of processes, and that really mutations sometimes are really just telling us about those underlying processes, and they might have a good one-to-one and onto mapping with them, but they're still kind of a surrogate for an underlying process, and there are processes that don't have single mutation surrogates. Okay, great. So, then how do we get here? How do we end up in this place? And that's what I'll do the last, 5 or 6 minutes to, kind of giving you a sense for where this might all start. And you know, I took the idea that, all right, if it's starting somewhere, it could be germline and it could be individual behavior. So let's start off with germline, and if that doesn't work out, then we'll start looking at individual behavior. And so we started looking at a couple of features of the germline genome, and I'll motivate quickly why this is a great idea in prostate cancer. Obviously, in multiple cancer types, there's lots of germline risk factors, but actually, I'll make a simpler argument. The typical prostate cancer, as I showed you, has 3,000 point mutations when localized, up to 30,000 when metastatic. Any two people on this call have at least, any two non-consanguineous people on this call have at least 3 million germline SNPs that distinguish us. So there's actually far more genetic variability in the germline than between tumors. Equivalently, even if we think about structural variants, prostate cancer is pretty quiet. So it has maybe 4 or 5% of the genome with a median structural variant influence. Well, it's 1% from the latest NOMAD data in germline population, so it's still pretty comparable. And in fact, we have BRCA2 carriers that have a highly elevated risk of diagnosis of prostate cancer and a 4 times higher risk of an adverse event if they get it. And there are polygenic risk scores, so made from common variants, that have very, very strong influence on telling us about prostate cancers that have a low risk of diagnosis or relapse. And so we started off looking at those germline BRCA carriers. We looked at a set of 20 of them, whole genome sequencing, and I'm just going to draw your attention to one thing up at the top. Here we're taking a look at MYC copy number aberrations in BRCA2 carriers. You can see that they occur in 75% of carriers, but only in 25% of sporadic cases. So clearly those germline-driven cases are a little bit different. I'm not showing you, but actually every single BRCA carrier that we looked at was a branched evolution, whereas it was mixed whether it was branched or not in sporadic cases. So germline might be influencing somatic evolution. That makes sense. Let's start off looking at common variants and the simplest place we could look, methylation, the epigenome. We did the somatic methylome, tumor methylome, and whole genome sequencing of the germline in 400 tumors. And this work was done by Katie Houlihan, a PhD student who's going to be joining you guys for a postdoc with Christina Curtis like in a couple of months. So Katie went ahead and looked at this and said, all right, is there an association? Easiest place to look is at germline risk variants. So she looked at a set of 120 germline variants and asked whether there was a specific CpG anywhere in the genome that was associated with it. And for about 20% of germline risk variants, there was, sometimes multiple that are not in linkage to equilibrium, and a couple of cases on different chromosomes. Okay, 400 tumors, you might not be totally convinced. So we went ahead and looked at this in a validation cohort of about 500 tumors, and you can see a little bit of moderation of the effect size, but basically everything validates. So we see strong germline somatic interactions at prostate cancer risk alleles. Risk alleles, sure. But what about, you know, methylation marks that are associated with patient survival? So Katie went ahead and took the 20 methylated CpGs in the genome, most associated with prostate cancer outcome. None of these are super good biomarkers, but you know, they're all associated with outcome. And she looked to see whether or not there was a germline SNP associated with them. For 19 out of 20, I think there were, and in some cases multiple. So you can see several cases, a single SNP would be influencing multiple CpGs. And that means that we've got germline SNP interactions that are associated with patient outcome. And again, these validated multiple cohorts. Okay, so now we've got this story where germline is influencing methylation in the tumor and patient survival. But is it methylation specifically in the tumor, or is it just prostate methylation everywhere and it affects normal prostate and tumor methylation? And so we did a germline screen to look at this. We have individual SNPs that are prognostic, like this one, and then we ask for every SNP in the genome, is there a methylated locus anywhere within, I think it was 50 kb up and downstream, which is correlated to its methylation status. This is a Manhattan plot of this. You can immediately see really high p-values, 10 to the minus 100. To give you a sense of what it looks like, you know, effectively the methylation is entirely determined by the SNP genotype. And then we said, are these tumor specific? So we required each of these to validate in an independent cohort of 500 at a genome-wide significant level. So that takes us down to about 7,000 of these. Then we required that these not show an association in 3 independent cohorts of normal prostate methylation, not show an association at 0.05. And that leads us to a set of about 1,100 tumor methylation quantitative trait loci. Some of these are associated with patient outcome. Many of these are also expression or protein quantitative trait loci. And you know, one other thing I'll show you is that they preferentially occur in enhancers, so they're in gene regulatory regions. Okay, so if prostate cancer shows germline epigenetic interactions, what about somatic mutations? So Katie went ahead and looked at 650 tumor whole genome sequences with both germline and somatic associations, and 600 for a validation cohort. And wouldn't you know it, for prostate cancer driver genes, that same set of 40, Katie identified 43 germline SNPs that were significantly associated with the chance that that tumor was going to have that specific somatic mutation. So to be really clear, a SNP that somebody gets at birth is predicting what is going to happen in a tumor 60 years later, and a specific mutation presence or absence. And just to give you examples of it, here are SNPs that are associated with a 2.5-fold increase of the risk or decrease in the risk of having a mutation in FOXA1, a common prostate cancer driver gene, that replicate in both cohorts. Or here's another SNP that's associated with RB1 loss, not just in prostate cancer, but also in breast cancer. We see others of these SNPs that are important in pancreatic cancer or ovarian cancer. And so we see multi-tumor type validation of this. And that means you can come up with a biomarker to tell a patient at age 0, if you get prostate cancer, this is the type of mutation that you are likely to get. They're not perfect. It's a 70% AUC, so not great, but remarkable that that accuracy is possible. And in fact, this is for TMPRSS2-ERG, which I mentioned before. TMPRSS2-ERG shows strong ancestry-associated influences, different rates in Asians, Africans, and Caucasians. And the SNPs that we identified here explain 30% of the ancestry influences, and it's only about 7 SNPs that I'm showing you the effect of. So actually, ancestry differences can be driven by a remarkably small number of germline polymorphisms. So what did I show you? Hopefully I showed you that you can develop robust biomarkers using multiple data types, that these biomarkers correlate with one another, that to do this stuff really well, you need to integrate germline and somatic and imaging. And another major arm of our work is now incorporating fluid biomarkers, and I suspect Thomas will tell you a lot about that joint work in January. And that's the next steps, you know, figuring out a clinical care pipeline and doing large-scale validations. Obviously, I said we because I did like none of this. This is a large collaborative team. This is a subset of the team. Rob Bristow was the co-lead with me for 7 years before he headed off to Manchester. Thomas Kisslinger and Hansen He lead the Toronto arm of it, and I'm working closely with Wei Wei Ye and Rob Reiter at UCLA now and developing another arm to it. And these are the people who actually did all the hard work. Thanks so much. I'd be happy to take questions if there are any.
All right, thanks, Paul, so much for such an excellent talk. And everyone, please either raise your hand or write your questions in the Q&A, and we'll make sure we get them answered. Thank you. Please go ahead.
Yeah, hey, it's very nice. So I have one question. This evolution germline, this methylation and all, is the age-related things, like what did you segment it by age?
Aruna, we're having a little trouble hearing you. Could you repeat that or maybe adjust your audio?
Okay, just hang on 1 second.
Let me ping back the question, and if this was it, then maybe you could give us a yes, because maybe I got it. I think you asked, are the germline SNPs that we're looking associated with age at diagnosis or age of the tumor relative to its evolutionary start as best as we can infer? Was that what you were getting at?
Yes, that's correct.
Excellent. So yeah, great question. We specifically looked at only sporadic late onset tumors. So the average age of diagnosis in our cohort is like 67. The youngest individuals are something like 50. That being said, so we didn't look at the full range. We did not see an association in our cohort, but our good friends in Hamburg at the Martini Clinic have a large cohort of about 250, I think, whole genome sequences in early onset, so men under the age of 40. And so we did look in that cohort, and fascinatingly, almost nothing matches up. Not even a little bit. It's remarkable. I think it was like 2 of the interactions showed up with very strong validation, and everything, there wasn't even a hint of it. And so that suggests, you know, that very early onset prostate cancer and late onset prostate cancer are in some ways fundamentally different diseases. And you can imagine all sorts of reasons why that might happen. It's a relatively smaller cohort. So, you know, we, in the paper we just got sent out a little while ago, is we undersell that point because, you know, it could be a series of false negatives, but my take on it is that there are pretty substantial age-associated differences. The other part, evolutionary age, to the extent that you believe the timing estimates that we do through work like we did in PCOG and in my team since then, there's no association, but I have healthy skepticism about the accuracy and error bars in our timing estimates, so hard to know about evolutionary age. We're doing a series of studies with longitudinal sampling that should be able to start resolving that next year or maybe the year after.
Great. We have another question. How many of these biomarkers are specific for type of treatment, radiation therapy or hormonal treatments?
Yeah, great question. So it's really frustrating. So we looked really intensely at biomarkers that might be good predictive biomarkers for surgery versus radiotherapy, and we can't find anything. And like, we have enough cohort to like be sufficiently powered for it and look at the interaction term. And, you know, it's a gigantic chunk of negative data that we invested a lot of time looking at. And there's just nothing there. If it's a poor prognostic marker for surgery, it's a poor prognostic marker for radiotherapy, and those are at clinical equipoise. And we couldn't find anything except for the well-known BRCA mutation characteristic, where there's reasonable data, although not quite convincing yet, that patients with a germline BRCA do worse with radiotherapy than they do with surgery. So, you know, you can imagine reasons for that. But even there, that's not quite sufficiently firm to be practice changing yet, although, you know, I think there's a number of cohorts looking at that now. So the long and the short of it is, no, in terms of hormone, the cohort's about 10 years old in median survival, which means that these patients are just relapsing now with metastatic disease. They had their local relapses or biochemical, but not a bone met that was observable. And now we're getting true bone and the first visceral metastatic lesions. And so we're not even close to powered to look at hormone therapy responses yet, and won't be for a few more years.
Great, and kind of a follow-up as well. He says, I would have felt that hypoxia markers would be associated with RT response. Yeah,
so did we. And so, you know, I mentioned Rob Bristow was my co-lead for that part of the work. And, you know, the number of times I heard that from a rad onc and was, are you sure you did this right? Because it should be associated. And, you know, there's nothing there. It's really frustrating. One of the things that we're exploring now is if we can use a different sequence in MRI imaging to be able to measure that. And a lot of people keep asking us about whether oxygenation might be beneficial in highly hypoxic tumors. And we're doing a little bit of work now with Lee Jones at MSK on exercise and looking at how exercise is reshaping the hypoxic landscape of prostate cancer. So hopefully we'll have some more stuff there, but at least in this setting, we're not seeing anything, no matter how we measure hypoxia or, you know, what kind of a cohort or selection we do for RT response.
Great.
So yeah, one more question if no one is asking, like, is it okay, Ryan?
Oh yeah, please go ahead.
Yeah. So there are twofold. The very first slide, very impressive things. I think something around 1969, the PCA marker, like a very short peak goes and then comes out. That's because of the false positive, or that's because of PCA recognized as one of the marker, and first time like they could able to diagnose more people, and that's why we could able to get short peak, number one. Or number two, what about the prostate cancer? Like, because it's most of the patients late 60s or something. Is that any hereditary related genetic connections are there? Like, I know this is out of things, but I'm just wondering, these patients are like any genetic background, I mean, hereditary background.
Yeah,
or
great question. So the first one was PSA testing. What's really happening there? So this is the best understanding of the field. Prostate cancers that are low-grade, indolent, not going to kill anybody are hyper-frequent. And if you take like a 95-year-old and do an autopsy specimen, something like 50% of men at that age will have a low-grade tumor in their prostate. But it's never going to kill people. It just never becomes aggressive and metastatic. And so PSA testing found this large surge of indolent tumors that are clinically insignificant and either lacked the ability to become metastatic or had such a low probability of becoming metastatic that it was not happening in an appreciable number of patients' lifetimes. So we see this big increase in incidence, but no corresponding increase in mortality because we're not saving people's lives. What we're doing is giving people the cancer diagnosis without, changing their lifespan ultimately. And in some cases, treating and causing stress and financial toxicity and stuff like that. That's the overall interpretation of the PSA peak for the field. And I take no credit for that work. That was way before me. The second part is, are there genetic factors for prostate cancer? So let's leave aside BRCA2 in particular. There's been a lot of recent work that rare DNA damaging repair gene mutations, deleterious pathogenic mutations in them, so ATM, PALB2, BRCA1, CHEK2, are associated with an increased risk of diagnosis and an increased aggressivity, so higher grade at diagnosis or higher risk of relapse after definitive local therapy. And there's a series of common germline SNPs, about 120, which have been put together in a polygenic risk score marker by a consortium called Practical, led by Raziels at ICR in London. And that polygenic risk score identifies, I think it's the top 2% of men with the least genetic risk have 5-fold less chance of being diagnosed with prostate cancer in their lifetime, and the 2% bottom have 5% more. So of course, that's not like most individuals, but it really shows that prostate cancer has a genetic risk component. And twin studies show that prostate cancer is the most heritable solid tumor, non-pediatric solid tumor with, I think, 57% heritability, H-squared of 57%. So yeah, there's a clear genetic component to that. Yeah, great questions.
Another question is, do you have some hypothesis for the survival difference between monoclonal and polyclonal tumors? I'm also asking because I've observed exactly the opposite effect in colorectal and pancreatic cancer.
Interesting. So yeah, I have a hypothesis. It goes like this. Every prostate cancer is polyclonal if we look at it spatially. Because we looked at biopsy specimens, I think what we're getting is something about mixing of subclones as opposed to spatial variation of subclones. And so when you see that in a relatively small tissue sample, those subclones have mixed, it's telling you something about the branching of the evolutionary process. And with highly branched evolutionary processes in prostate, I think that makes it more likely to escape from the prostate, because that's ultimately like the determinant of lethality and aggressiveness. If it's localized, then definitive local cuts it out, so it doesn't matter. And so the hypothesis is mixing is telling us something about ability to A, get out, and B, establish in a metastatic niche. That's my hypothesis. I have only like correlative data for that. It's a guess. I would speculate on the pancreatic side. that the reason why monoclonal is more tightly associated is probably because the time between the most recent common ancestor and diagnosis is so short. And in prostate, it's so long. So I would guess that like pancreatic cancers come from a burst of a suddenly highly proliferative evolutionary aggressive subclone, and that those are rapidly proliferating and more aggressive. And by that time, they've already escaped the pancreas, and pancreatic cancer is difficult enough to manage, and that's kind of the origin of it. Speculation entirely on pancreatic. I don't do that much pancreatic genomics yet.
Great. Another question is, so crazy that non-mutated tumors can still potentially be so aggressive. Are these tumors AR-driven or demonstrate neuroendocrine-like features?
No neuroendocrine features that we could see. Great question. In fact, they histopathologically look indistinguishable. So the way we did it is we took, I forget, it was 10 or 15, and we took 10 or 15 matched by every clinical feature that we had. And then we told the mix of 20 or 30 in total to our pathologist and said, look at these with everything you can think of and see if you can find a way to separate them. And he came up with lots of things that separated things, none of which correlated with the evolutionary noise. It was like, we looked at T cells and a bunch of different stains. And yeah, basically there was nothing that we could dig out of it, which is weird and surprising. Are they AR dependent? Tough to know 100%, but just on the basis of the subset of patients that have had relapses, they seem to be ADT sensitive in a similar way to the rest of them. That's anecdotal rather than a well-powered statement at this stage of the cohort's evolution, but it doesn't look like we're seeing differences in interval between biochemical and metastatic relapse or in responsiveness to first line. We'll see what happens when they go on second and third line, so Enza and Abby and stuff like that. So no obvious triggers there that we could see. So we've been trying to get funding to super deeply dig into that. And, that's one of my surprises is that a grant focusing on that has never gotten funded. So I think it's one of the most interesting things that I showed in the presentation, how that happened. So I'm with you.
Excellent. Any other questions for Paul? Okay. I was pausing to give Con the last word if you'd like. Otherwise, it was.
Thank you so much for this wonderful presentation. Maybe the next time we would love to see you come and visit us in person. We look forward to those days. It's a wonderful presentation. Thanks so much.
Thank you, guys. It's great talking to you. If anybody has questions, just reach out to me by email, and happy to answer other stuff. Bye, guys.
Thank you.